Hey everyone, welcome to our first experiment for the Gray Matter project sponsored by Yale. So in this experiment, we're going to be connecting to topics such as osmosis and sodium-potassium pumps, which were both covered in our last lecture given by Leah Harmon. But before we jump into the experiment, we got to get our safety on. So for this experiment, you're going to need 2 beakers that are at least 400 milliliters, 50 milliliter graduated cylinder, a cup for mixing, salt, food coloring, 10 to 15 centimeters of dialysis tubing per group, scissors, and distilled water. So for our first step, we're going to be cutting dialysis tubing roughly 10 to 15 centimeters long. And then once you have that cut out, we're going to be soaking it in the distilled water for roughly 15 minutes. So you're waiting for your dialysis tube to soak. You're going to need to prepare a saline solution. So what you're going to need for that is about 30 milliliters of distilled water, which I have prepared here. So you're going to pour that into your mixture cup. And you're going to continuously add salt until you cannot dissolve anymore. If you just so happen to add too much salt to your solution, you can go ahead and dilute it more with extra distilled water. So 15 minutes later, you should be able to take your dialysis tube and rub your 2 fingers against it so that it forms an opening. But before we do that, we're going to have to close off the dialysis tube by forming a knot right at the end. In order to make it easier to put the solution inside of the dialysis tube, we're going to put it right back in the graduated cylinder. And so then what you're going to do is you're going to get the dialysis tubing out of the beaker, rub your 2 fingers against so that you can make an opening. And then, once you have an opening, you're going to go ahead, take the solution, and put it inside of your tube. And then once you have enough, what you're going to do is you're going to knot it off right at the end. So for the next part, you're going to take your other beaker, and you're going to fill it with distilled water, just enough so that it can cover the dialysis tube. Then we're going to add a couple drops of food dye. Give it a stir. And then you're going to take your dialysis tube and add it to the solution. Make sure you are covering it. Just a side note, your dialysis tube should be fully submerged in the solution. Then after that, you're going to wait about 20 minutes. Now don't be alarmed if yours does not look like this because ours has been sitting out for about an hour. But you will notice that the dyed water moved from outside to the inside of the dialysis tube. Now stay tuned to learn about the science behind it.
In biology, cells are surrounded by a semi-permeable phospholipid bilayer. In the experiment, the dialysis tubing represents this membrane. Aquaporins are the integral membrane proteins that allow water to pass through the membrane through a very specific form of diffusion known as osmosis. Osmosis can be defined as the movement of water from an area of high concentration to an area of low concentration. For our purposes, this can be defined as the inside of the cell, and this can be defined as the outside of the cell. There are 3 types of osmotic environments that a cell can be placed in. The first type is very similar to our experiment. Inside the cell, there is a very high concentration of solute, and outside the cell, there is a low concentration of solute. Conversely, it can be seen that there is a high concentration of water molecules outside of the cell and a low concentration of water molecules inside. Because the water molecules travel from areas of high concentration to areas of low concentration, water molecules will travel into the cell. We can say that the outside of the cell is hypotonic to the inside of the cell. The second type of osmotic environment involves a high concentration of solute outside of the cell and a low concentration of solute inside of the cell. This means that there is a low concentration of water outside of the cell and a high concentration of water molecules inside of the cell. In this situation, the water molecules will travel outside of the cell. We can define the outside of the cell as hypertonic in relation to the inside of the cell. The last type of osmotic condition involves equal amounts of solute inside and outside of the cell. The water molecules will be at something we can call equilibrium, meaning that there is no net movement in or out of the cell. We can define this as an isotonic environment. In neuroscience, diffusion is essential to the propagation of action potentials down an axon. In the case of the sodium-potassium pump, sodium and potassium ions need to be restored back to their resting potentials following the propagation of an action potential. This type of diffusion is known as active diffusion due to the addition of energy in the form of ATP to the exterior of the pump. When the sodium concentration is extremely high outside of the cell, 3 sodium ions bind to the inside of the cell pump, and ATP phosphorylates the sodium-potassium pump. This addition of phosphate induces a conformational or shape change in the pump, allowing 3 sodium ions to be released to the inside of the cell and for 2 potassium ions to enter the pump. The phosphate group is then removed, initiating a second conformational change, allowing the potassium ions to be released to the cell's external environment. These restored concentrations allow the axon to return to resting membrane potentials, preparing itself for the propagation of a new action potential. Thank you guys so much for watching, and stay tuned for the next episode.